pH of SDA agar. Option A 4.1, option B 7.2, option C 5.6, option D 5. Answer is option C 5.6. Vaccines are sterilized by option A hot air oven, option B autoclaving, option C sage filter, option D heat inactivation. Answer is option D heat inactivation. TC cells are restricted. Option A MHC class 1, option B MHC class 2, option C MHC class 3, option D MHC class 4. Answer is option A MHC class 1. Spirochetes are stained by option A gram stain, option B India ink, option C Levedity stain, option D Leishman stain. Answer is option C Levedity stain. Killing reaction is seen with option A pneumococcus, option B streptococcus, option C staphylococcus, option D gonococcus. Answer is option A pneumococcus. Immunoglobulin which crosses the placenta. Option A IgA, option B IgM, option C IgG, option D IgE. Answer is option C IgG. Rapid diagnosis of tuberculosis is possible with option A Oramin rhodamin stain, option B Seal-Nielsen stain, option C Giemsa stain, option D Gram stain. Answer is option A Oramin rhodamin stain. Transport medium for stool sample in cholera is option A thioglycolate, option B Dubose medium, option C Carrie-Blair medium, option D selenite F broth. Answer is option C Carrie-Blair medium. Which of the following interact with the macrophage and secrete cytokines? Option A cytokine T cell, option B helper T cell, option C B cells, option D NK cell. Answer is option B helper T cell. The primary target to HIV infection are option A Th cells, option B TC cells, option C macrophages, option D NK cell. Answer is option A Th cells. Holding period of hot air oven is option A 160 degrees Celsius for 60 minutes, option B 121 degrees Celsius for 60 minutes, option C 121 degrees Celsius for 15 minutes, option D 160 degrees Celsius for 15 minutes. Answer is option A 160 degrees Celsius for 60 minutes. Cutting instruments used in ophthalmic surgery are sterilized at option A 160 degrees Celsius for 2 hours, option B 160 degrees Celsius for 60 minutes, option C 150 degrees Celsius for 2 hours, option D 150 degrees Celsius for 1 hour. Answer is option C 150 degrees Celsius for 2 hours. Oils and glycerols are sterilized at option A 150 degrees Celsius for 1 hour, option B 160 degrees Celsius for 1 hour, option C 150 degrees Celsius for 2 hour, option D 160 degrees Celsius for 2 hour. Answer is option A 150 degrees Celsius for 1 hour. Older method milk is heated at option A 100 degrees Celsius for 5 minutes, option B 63 degrees Celsius for 30 minutes, option C 55 degrees Celsius for 30 minutes, option D 72 degrees Celsius for 30 minutes. Answer is option B 63 degrees Celsius for 30 minutes. Which antibody serves as an antigen receptor for B lymphocytes? Option A IgA, option B IgG, option C IgM, option D IgD. Answer is option D IgD. In flash process, milk is heated at option A 72 degrees Celsius for 15 to 20 minutes, option B 63 degrees Celsius for 30 minutes, option C 55 degrees Celsius for 30 minutes, option D 72 degrees Celsius for 15 to 20 minutes. Answer is option D 72 degrees Celsius for 15 to 20 minutes. Dash is the equipment used for sterilization of LJ media. Option A incinerator, option B inspissor, option C autoclave, option D none of the above. Answer is option B inspissor. Tyndallization is option A 160 degrees Celsius for 20 minutes on 3 successive days. Option B 100 degrees Celsius for 20 minutes on 3 successive days. Option C 80 to 85 degrees Celsius for 30 minutes on 3 successive days. Option D 80 to 85 degrees Celsius for 60 minutes on 3 successive days. Answer is option B 100 degrees Celsius for 20 minutes on 3 successive days. Concentration of blood used in blood agar is option A 10 to 20 percentage, option B 5 to 10 percentage, option C 10 to 50 percentage, option D 2 to 4 percentage. Answer is option B 5 to 10 percentage. Which are the two types of agar? Option A Australian and Japanese, option B Australian and New Zealand, option C New Zealand and Japanese, option D Canada and Japanese. Answer is option C New Zealand and Japanese.